Hey guys, welcome back to Python/Linux. In this lecture, we're going to be continuing to work with our pipes and filters. There's a part 1 video, so if you are watching online and you didn't do the part 1, then go back and watch that first, and then we'll continue on with this one. So let's go ahead and jump right on in. Okay, so this is where I kind of left off. I'm going to clear. Alright, so just for your reference, print working directory, this is where I am, in the proteins folder again. So what if I want to do 2 commands at once? I'll give you an example. So we have our lengths .txt, right? And we have our sorted lengths .txt, right? What if we want to do a sort and a head function together? So, for example, we want to know what the shortest length is in our protein, the number of lines in our PDB files, right? So we did that word count star dot PDB, right? And so we got our lengths, and then we did the sort function where we said, okay, let's sort them from smallest. But what if all we want to know is the shortest length of lines in all of these files in this folder? What we can do is we use sort -n, which we did before, lengths .txt, and then we do this pipe function, which is above the enter. It looks like 2 arrows or 2 dashes going vertically. On my keyboard, at least, it probably depends on your keyboard. And then we say head -n1. And so what we're doing is we're saying, okay, sort the lengths of the proteins and then just give me the top one, right? So it's 2 different commands, the sort command and then the head command. It says do both of these at the same time. And so from that, we say that 9 is the shortest number of lines for one of these files, and it's that methane file. So we essentially can pull that information out without having to create a file like we did with this head, or with this, let's do less sorted lengths, right? So all we wanted to know was what the smallest file name was. And so we can do that with this command instead of saving things to files and things like that. Okay, so let's clear this. So you can do as many pipes in a row as you want. You probably don't want to get too crazy, because if one breaks, then the following ones, or like if one is incorrect, the ones further down the line probably aren't going to work. It'll just be a big mess trying to troubleshoot. But let's do this word count L for our protein database files, and then pipe sort n. And so as you can see, we pulled just the line numbers and sorted them all in one command, as opposed to previously we had line numbers and the words and what was the characters, right, all together. So if you just want to pull just line numbers, have them sorted, we can put those 2 commands together. So we can also send the output through another pipe. So let's do line numbers of all the protein database files, and we'll sort them like we did in the previous command, but we'll also just print out the head, so the first one. So we've now created a list of all the line numbers for the protein files. We then sorted them from smallest to largest, and then we printed out just the top one. So this is kind of a cool figure that they provide to show what's happening here, or how some of this works. So here we have our word count giving lengths, and it outputs into the shell. If we do this word count, the same function, but then we use this greater than, less than symbol to put it in a file called lengths. It puts it into a file, so the output's a file and not the shell. And then if we do these pipings, so we do word count or the line count for the protein database files, and then we sort them, and then we head them. We do an output of shell again, because we didn't do this this part here. But if we would add this part to the end of this, it would do all three functions and then put it into a file. Oops, here's my Detroit Lion news in between recording videos. A tortured Lions fan. Okay, so linking these programs is what makes this Unix environment, which is the underline of Linux, very successful. So instead of creating these giant programs, what it does is creates these little building blocks that you can it's kind of like Legos. Like, you can pick little commands and arrange them and put them in certain orders to get the output that you want for your navigating your directories or your bash script, if you're using a bash script. So it makes it very intuitive, or very easy to use once you learn these little building blocks to kind of put things together in a way that makes sense to you and helps you to move files in the right places and run the right Python scripts and things of that nature. So let's see. Let's all right. So let's get back to Nell's conundrum that we were, or project. Oops. Oops. Oh. I just completely logged out of my thing here. Sorry, we're at a time to take a break. Good time. All right. So let's get this going again. All right. Let's open my terminal. All right, so we want to go back to sorry, I'm bad at typing and talking at the same time. All right, let's go back to the shell lesson data, and we want to go to this Nell's data. So this is the North Pacific gyre, right? And so she's created 17 files, all these text files, in this directory. So if we were to type wc-l txt. For this, you can see that we have 5,000. We have 18 lines with 5,040 total in the folder. Or we have 18 files, I should say, with 5,040 total lines in it. So if we do wcl.txt and then we pipe, we say sort-n, and we pipe, and we say head n5. What we get is we've sorted by number, so smallest number, and we got all the line numbers for all the text files, and we display the top 5. So you can see that for some reason this 2018 only has 240 lines instead of 300 like all the others. So part of this working problem says that null goes back to look. So this is kind of like an error checking step, right? So null goes back to look and notices that these 60 lines that are missing from this file right here are probably because that she forgot to assay at 8 o'clock on Monday morning. Someone was probably using the machine on the weekend and she forgot to reset it. And then she checks also to do dash.txt. We want to see if there's too many lines, right? Sort and so instead of head, we use tail, right? So it's the last 5 lines of the file that's sorted. And as you can see, none of these have too many. It's not like 600 or something like that. So this is the bottom of the list here, right? And so, but she does notice when looking at this that we should have A and B reps, but she has a Z. And so all of her samples should be marked A or B, but her lab uses Z to indicate samples with missing information. So if she wants to find all the Z samples, she ls anything z.txt. So this, remember, asterisk means anything. So anything and then the letter z.txt. So let's look at what files. So it appears we have 2. 1971Z and 2040Z. So it says she checks the log of her laptop and there's no depth recorded for these samples, and it's too late to get the information any other way, so she must conclude those 2 files, or must exclude those 2 files from her analysis. She could delete them, use them RM, but there are actually some analysis she might do later where depths don't matter, so instead she'll have to be careful later on in selecting files using the wildcard expressions. She can do this. Since they all start with mean, and then she wants anything a.txt, right? So this lists all the files that start with mean, then have any number in there, and then end with a before txt. And similarly, you can do the same thing with mean asterisk b.txt, and I'll list everything that starts with mean. Right? Okay. So we're going to not delete those files. You could delete those files by doing the rm function that we talked about earlier, but we're going to leave them there. But that's it. So that is piping. So it gives you an idea. There's a lot more you can do. If you're like more of a software engineer or something, you might get more into some of these commands like I said, for the most part, there are some things that I've done in Bash or in the terminal. Scripts that I've run when it's just easier than writing Python because you don't have to import packages, or it's just faster to just write a Bash script. So I have a lot of stuff that I do for my microgravity research, for my space biology stuff. I use a lot of small motors and things like that to rotate plants, and now mushrooms. I've expanded into mushrooms, but to rotate these things in 3 dimensions to kind of cancel the effects of gravity. But I use stepper motors, and to control those stepper motors, I usually use Bash scripts, just because it's quick and easy, and it's just a few commands. You could use Python, but you have to import a bunch of packages into it. So there are some things you can do with Bash. Anyways, I'm just kind of rambling on Bash. But the best thing is just to be able to know how to get everything in the right spots for you to run Python scripts. That's kind of the big thing. Or to navigate. When you do any sort of cloud computing, you're going to have to know how to use the terminal like this, because those are generally all Linux environments. So you're going to have to move your stuff into the cloud and into the right folders for you to execute your Python scripts. So knowing piping and things like that are also important for that, and also for error checking, like we did with Nell's data, the Z stuff, or the file that's 240 lines instead of 300. You might have your analysis network, and you're going to have to figure out why. And so using the terminal to look at all your input files, to look at your Python scripts, to look at everything to see that it's all correct, is kind of an important tool to have in doing this. So anyways, that's the end of pipes and filters. I think we get into loops next. So I hope you enjoyed the video. If you're watching on YouTube, or if you're watching and it's just in class, you guys can hit like and hit subscribe too. But I appreciate it. I want to get this out there. I think this is something important that everyone should know, because this is where biology is going. It's going to be less about doing PCR and more about doing whole genomics and things like that. But I'll get into that diatribe later. Hit like, hit subscribe. We'll continue on our Linux journey and our command line journey in the next video. And so I'll see you in the next one.